Hello, my name is Shana Arias Garcia, and I go to William Patterson University. I will be presenting my research on the examination of the central and lateral amygdala in the BTBR mice model of ASD. The control group I've chosen for this research is a commonly used inbred strain known as C57BL67, also known as C57 for short. This strain of mice are used in a wide variety of research, including cardiovascular biology, developmental biology, genetics, and immunology, so forth. So the C57 mice were chosen because they bred well, they are long-lived, and have a low susceptibility to tumors. The BTBR mice, or their full name, BTBR T supersubscript plus ITPR3TFJ, were obtained from the Jackson Laboratory, where they were inbred for at least 20 generations. They have 5 important genotypes: the ITPR3TF, which means they have waves of hair loss and growth; the A supersubscript plus, which gives them the distinctive black and tan coat; the DISC1 deletion, which is a 25 base pair deletion that causes a frame shift and a premature termination of translation of proteins; the T supersubscript plus, which is also known as bronchiary. It aids in development of the neural core during embryogenesis, which helps control the development of the neural tube. And the neural tube is the layer of cells that ultimately develop into the brain and spinal cord. Lastly, they have the COX7AL superscript L gene, which is an allele that encodes the long isoform with 113 amino acids. Autism, also known as ASD, is a spectrum disorder which affects socialization and communication. The causes of ASD are not fully understood, but it is believed that abnormal development of the brain structure and/or exposure to environmental factors may play a role in its occurrence. Since it affects everyone differently, we can't exactly pinpoint one specific thing to determine autism, but some noticeable behaviors seen deal with repetitive and restricted behaviors. The BTBR strain of mice exhibit symptoms of autism, such as reduced social interactions, meaning we don't see them mate as often as you would with other strains of mice. They have low exploratory behaviors, unusual vocalizations, high anxiety, meaning when we come near them, when we touch them, they start to get nervous, and this is where you start to see the symptoms I mentioned prior, where they'll start to repetitively groom themselves, and it causes blotches of hair loss throughout their body. The region of study is the amygdala, which can be found within the temporal hemispheres and is divided into central and lateral subnuclei. The central amygdala is understood to control body responses to fear, stress, and anxiety, whereas the lateral amygdala plays a part in its input sensory. I hypothesize that while observing cell differentiation in the amygdala, I will find a disparity between the central and lateral amygdala between the C57 and BTBR mice. To cut it short, the procedures we went was perfusion, which is a technique used to take out their brain carefully so we can put it in PFA for 2 days and then switch it over to sucrose until needed. That brain slice is then taken out and microtubed, where it's cut into very thin slices. After that, those thin slices are mounted onto slides, where those slides are stained with crystal violet. And once dry, we move on to imaging, where we zoom into the brain slide and we start to take individual photos where we upload into Photoshop and piece together like a puzzle. After it's pieced together, we use image J to measure the area of study. So I included the data basically that I collected. The top part shows the imaging, 3 sections: the anterior, the medial, and the posterior. In the anterior, you can see that there is no real development of the hippocampus yet, which is the middle section near the little hole. And then the second image shows the medial section, which you can start to see that the hippocampus is starting to develop. And in the third, you see the hippocampus takes up most of the image. After I measure all these areas, I basically split it into 3 sections: the anterior, medial, and posterior. These measurements were then separated based on location, either left or right. And throughout the data, you can see that there are certain parts that show NP, meaning that in that image at the time, the area that I was looking for was not present. Once put together, I made the graph that shows an average of the 3 sections, and then an average of, the average of each individual section, and then the average of those 3 sections to get an average of the central and lateral amygdala. And then it basically shows a side-by-side comparison. To conclude, the central amygdala is known to play a role in the fearful and stressful stimuli, as well as anxiety. The data obtained from this research show that central amygdala was larger in C57 with an average of 868,026 when compared to BTBR, who showed an average of 842,022. The lateral amygdala receives information and the basolateral processes it before sending it out. The lateral amygdala was smaller in the C57 with an average of 129,443 when compared to the BTBR, who showed an average of 141,204. The lateral amygdala receives information and the basolateral processes it before sending it out, as I mentioned prior. The differences between the amygdala and the control and experimental group were not as significant as I hypothesized, and I plan on gathering more data and increasing my numbers in order to help improve the statistics in the future and see if I can get a greater disparity between one or the other, or maybe notice that there is much of a difference. I would like to thank Dr. Lee for being my mentor, Marisol Mina for being the animal technician at William Patterson and helping and guiding me through the process of working with mice, GSLAMP for funding my research, and Dr. DeRoche for giving me this opportunity. Thank you and have a wonderful day.